Good morning, and thank you for joining us for our webinar, On-Site Environmental DNA, The Future is Here. My name is Mario Thomas, co-founder and CEO of Precision Biomonitoring, and your host today. Joining me is Mark Wilno, wildlife biologist. Environmental DNA, or eDNA, is a relatively new science that has started to revolutionize industry approaches to biomonitoring. What we are presenting today is our most important initiative, an innovative service providing real-time eDNA results at the point of need, right in the field, offering game-changing benefits in terms of time and cost savings. We appreciate this opportunity to discuss how eDNA can benefit you, and have strived to keep this webinar concise and practical. Firstly, I wish to express our sincere thanks to Stantec, Azimuth, AECOM, Parks Canada, Ecometrics, and all the collaborators who have helped us build and validate this exceptional platform developed at University of Guelph, and to launch it commercially last year. Our agenda this morning will be a 30-minute presentation followed by a question period that includes the following: the challenges of conventional biomonitoring, a primer on environmental DNA, assay design and why all assays are not equal, a paradigm shift in the eDNA process, and how excellent science drives reliable results. At this point, I will invite our co-host, Mark, to dive into the most interesting aspects of our 5 topics.
Good morning, and thank you, Mario, for the introduction. As you know, the conventional approach to biomonitoring involves capturing, sampling, handling, and visually confirming target species. It is typically characterized by logistically challenging multiple visits to study sites that are both labor-intensive and commonly constrained by target species life stages and the impact of seasonality. As a result, these conventional survey methods may risk disturbing the habitat and harming the target species, potentially producing uncertain results. Challenged with the practical limitations of conventional biomonitoring, scientific research has steadily increased over the past 10 years into the potential utilization of molecular diagnostics. This takes us to the second item on our agenda for this morning, a primer on environmental DNA. eDNA is a groundbreaking technology. This young science allows extremely small traces of DNA constantly shed by organisms through bodily fluids, skin cells, or other secretions to be detected in water samples without having to directly see and collect the organisms themselves. So what is the fate of eDNA in water? Due to several elements like temperature, hydrolysis, low pH, and UV light, eDNA is degraded and will reach an equilibrium. If the target species is removed from the environment, eDNA will not be detectable after a few days or weeks at the most. This means that if a trace of target DNA is detected, the species of interest is either present or has been present very recently. It is worth being familiar with the rapid advancements in this field of DNA, beginning with the development of the method for nucleic acid replication in 1971, followed by the development of the PCR or polymerase chain reaction method, which was recognized with a Nobel Prize in Chemistry in 1993. While it was another 15 years later before the initial eDNA referee journal publication by Fisitola in 2008 on the invasive American bullfrog in European Alps, the pace of accelerated growth in environmental DNA was marked with more papers published in 2018 than in the previous 10 years. We must underscore that in 2009, The MICE guidelines, as they're known, were first developed for qPCR reporting in clinical chemistry as a minimal mandatory standards for eDNA qPCR assay design and optimization, in which one might expect the target DNA to be even more ephemeral or at lower concentrations in the environment as compared to cells or tissue samples. The most recent advance occurred in October 2018 when the University of Guelph hosted the first national eDNA workshop entitled Pisces. In 2016, scientists from the Biodiversity Institute of Ontario at the University of Guelph founded Precision Biomonitoring. Initially based on the Barcode of Life data system, or BOLD, these scientists developed a proprietary eDNA method known as the triple lock platform, providing highly specific and sensitive tests. Pilot testing and third-party validation of our eDNA tool has been completed with the generous support of Stantec and other visionaries, including Dillon, Azimuth, ACOM, Parks Canada, and Ecometrics. When done correctly, meaning not just grab and go, the most obvious advantages of eDNA are its high sensitivity, the speed of the process, and the resulting lower cost compared to conventional methods. As you can see, the list of potential applications of eDNA keeps growing with experience. So far, we have leveraged the benefits of eDNA in the following applications: for the presence or absence of endangered species, for early detection of invasives, for monitoring in remote locations, for the identification at early life stages such as eggs or larvae, and for the elusive or cryptic species, and of course, an all-time favorite, under ice sampling. Now let's turn our attention to the designing of eDNA tests. We want to emphasize that not all assays are created equal. Developing a reliable assay demands much more than creating just any primer and probe. The basic process is comprised of 4 steps: the in-house sequencing and/or accessing of external sequence databases, the aligning of DNA sequences from the target species and those from closely related species. Primers are designed to detect only the target species. Third, using different softwares, the molecular scientist designs primers and probes. And lastly, the best primer and probe are selected for optimization and validation in the lab using genomic DNA. Simple enough, right? While scientists at Precision Biomonitoring were not satisfied with this basic approach and developed the triple lock method for improving further the specificity of an assay. This piece of intellectual property was the basis for the creation of our company and is managed as a trade secret. Without getting into too many details, this is our key differentiator number 1, better assay tests. Rigorously developed and validated qPCR tests. Once primers and probes are designed in silico, they are validated in the lab against genomic DNA of the target and closely related species for maximum specificity. Sensitivity and efficiency are then optimized following verified standard operating protocols based on mandatory minimal standards adapted from the MICE guidelines. Lyophilized test kits are then manufactured, and we proceed with rigorous field validation using confirmed positive and negative sites. Precision Biomonitoring's third-party validation is accredited by the International Organization for Standardization, the ISO 17025, which is one of the most stringent certifications for testing laboratories globally. This accreditation indicates that all laboratories and personnel within the facility have demonstrated technical competence and the ability to produce precise and accurate test results. What Precision Biomonitoring offers is a true paradigm shift. The old model of shipping samples of water or filter papers to a lab is fraught with risks when it comes to precious picograms of eDNA. For starters, it can be unpractical to ship samples from distant locations. In addition, shipping increases the risks of quick eDNA degradation in transit. While many labs claim they can perform eDNA identification, uncertain quality control and method standards add to the risks. And then there is the time lag waiting for results. This old model is a barrier to adoption of eDNA. Precision biomonitoring reduces the number of steps that increase the risk of contamination and that of sample degradation from 5 steps to 3. The ANDI water filtration system and eDNA collection tool standardizes this crucial step with exact volumes, sample to sample, even flow rates, and single-use filter cartridges designed for reducing contamination risks. The filter containing the eDNA is removed and inserted into a vial with lysis buffer for the next step. The 3-minute eDNA extraction demonstrated to be on par with a 12-hour lab extraction. Once the purified eDNA extracts have been obtained, they are added to a PCR tube loaded with the target assay, a master mix, and a synthetic internal positive control, essential to assess the potential for the presence of PCR inhibition. The qPCR analysis is performed using the handheld battery-operated qPCR. The new version can simultaneously run 27 reactions thanks to 9 wells and 3 fluorescence channels. Brought together, these 3 steps make the Precision Biomonitoring integrated field lab. Wi-Fi and cellular connectivity make it possible to send the results from the study site to the cloud for data management. Strict and up-to-date protocols are followed to deliver high-quality results. Our process follows a quality assurance and quality control plan to ensure that cross-contamination of samples during analysis is minimized and verified at each step. This type of high-standard laboratory service ensures that our customers can be confident with the accuracy of their results. The main benefits of this innovative platform include time and cost savings, fewer visits, more rapid results, reduced personnel and equipment requirements, cost savings of 25 to 68%, field-ready components, on-site processing, multiple species per site, preservation of sample integrity, and the simplicity and safety reduction involved. In the last section, we will discuss how excellence in science drives reliable results. As mentioned earlier, reliable results are a lot more than grab and go. Detecting a target organism is probabilistic. In conventional ecological studies, this probability of detection depends on 3 types of factors: biotic, abiotic, and methodological. In eDNA studies, the probability of detecting eDNA in a sample is dependent on 2 major factors. First, the probability of capturing eDNA in a water sample, and second, the probability of amplifying an eDNA molecule from DNA extraction. By optimizing both, we increase the statistical power of the study. Statistical power in ecology relates to the ability of a survey or analysis to discriminate between the true presence and true absence of a species of interest. Power is predicated on the ability of the sampling design and the assay sensitivity and specificity to detect a target. Many factors will affect the probability of capturing eDNA in a water sample. Factors such as population dynamics, water quality, and flow rate will help decide on the sampling strategy, including the number and sites of samples, the volume of the samples, and the time of year. Our core key differentiator number 2 is our survey design methodology, on which we have prepared a provisional patent and for which we are busy developing a software tool that will standardize the customization of surveys. Each study deserves its own tailored survey design. As you can see, we currently include in our eDNA survey service close to 30 validated tests and have another 20 at various stages of development and validation. The benefits of environmental DNA, and specifically for the point of need or on-site eDNA tool, are pronounced, with overall cost savings ranging from 25 to 68% versus conventional methods. The field-ready eDNA survey services of Precision Biomonitoring offer significant benefits to the environmental consulting industry. Let's look at some specific comparisons to consider. We sat down with a varied group of consulting members to compare the cost impacts of using conventional methods versus using our Precision Biomonitoring eDNA surveys in 6 different client situations. You can see down the left-hand column that the sites, as well as the target species being assessed, were both applicable and typical for most Canadian environmental consultants. With the project questions being identical, whether using conventional methodology, the traditional approach, or the environmental DNA service of PBI, the cost savings with eDNA ranged from 25% up to 68% in these 6 situations. In conclusion, Precision Biomonitoring can provide you with the full eDNA survey services. With the one-stop shopping approach, our field scientists will consult with you on the customized survey design unique to your requirements, complete the on-site sampling, the eDNA extraction, qPCR, data analysis, and reporting. This all faster and at less cost than conventional methods for invasive species, species at risk, and keystone species, and not even withstanding the possibility of terrestrial or airborne eDNA detection. Back over to you, Mario.
Thank you very much, Mark, for the presentation. We would like now to invite you to include PBI's eDNA survey services in your 2019 field projects. And we would welcome the opportunity to discuss projects and specific timing with you. Thank you for your participation. We are not done yet. We would like now to invite you to ask any question you may have by clicking on the question button below the slides and writing them down in the space below. Also, by clicking on the poll button on the screen, you can vote on the topic you would recommend to us for our next webinar. So to answer the questions that you will bring up, I have Dr. Stephen Crooks, who is our head of development at Precision Biomonitoring, who will provide answers to your questions. So one question that I have, Steve, is how do you perform eDNA surveys for SAR bat habitat?
Good morning, everybody, and thank you, Mario. That is a very good question. I know from some experience, my partner is a bat ecologist, and some of the species in Canada do frequent water courses for feeding migration, and they do feed on the insects above them just on the surface of the water, and some drink from the surface of water bodies and pools. So we could conceivably take water samples from those pools and check for eDNA that's been shed from the saliva of these bats. One of the other ways in which we can use environmental DNA to monitor for bats is to monitor which species use maternity roosts, which is obviously of importance in managing these populations, and overwintering roosts. Now that populations in North America are subject to the plague that is a white nose syndrome fungus. In such routes, we could conceivably take guano samples, perform on-site eDNA extraction, and in essence, perform the same workflow that we do for water samples.
Thank you very much, Steve. The next question from our audience is from Kristen Beauchamp. And Kristen is asking, would multiple sampling of the same feature on different days reduce the potential for false positives?
Thank you for the question. That is absolutely true. When we're designing our optimal sampling strategies during our pilot phases, where we're doing eDNA validation in the field and in all real surveys going forward, we do use iterative sampling design. In that way, in combination with species occupancy models, that is a good way to keep in check these sources of potential error.
Thank you, Steve. I would like to mention a comment provided by Chris Wilson from MNRF on this point. Chris has commented that multiple sampling is very beneficial for habitat occupancy analysis. It doesn't alter the potential for false positives, but does reduce the risk of false negatives and increases the probability of detection, as we have described. Thank you for the comment, Chris, and Kristen for the question. So the next question is from Jody, asking how expensive is our setup. I will answer this this question. So each study, each survey is unique. This is why we do a survey design. We estimate what is required for this specific study. So we have developed and continue to fine-tune a methodology for tailoring a design that includes a sampling strategy for each study. So Mark showed you a range of costs for various scenarios. So the short answer to your question is that for each project, what we recommend is to do a short pilot study that will give us a chance to evaluate a number of variables that will influence the probability of detection, and then provide you with an estimate, a quote, for this specific study. So I hope that this answers your question. So the next question that we have is from Stuart Gibson. Stuart is asking, have you had any feedback from reviewing agencies, MNRF, regarding their confidence in this methodology? Will they accept this in lieu of their protocols? So the answer to this question is that in our experience, it has been a diverse feedback depending on district and regions. I mentioned Chris Wilson, who is on the science group, leading the science group on eDNA at MNRF. So it is building confidence at the moment. So we are at this stage where with new assays, new studies, we are building case studies with an overall purpose of building confidence with MNRF and also with DFO and conservation groups and municipalities. The next question is, which SAR turtles have assays, and has this method been applied to Ontario? So I will let Stephen answer this question.
Hello again. Thanks for the question. We have applied our on-site detection to 2 species of Ontario turtle so far. the Blandings Turtle and the wood turtle. The Blandings Turtle surveys took place largely at 2 different times of the year. We did overwintering habitat surveys for brumating turtles. So we went up to known occupancy sites where we had data from telemetry and direct observation, working with colleagues at Scales Nature Park in Aurelia and in the MNRF. And we got very high levels of confirmation of eDNA detection with known occupancy of brumating turtles. And this is, you should bear in mind, at a time of the year when the turtles are not shedding very much DNA at all because they have very highly depressed physiological activity. We've also done summer surveys of turtle activity in Nova Scotia, where there is a highly divergent population of Blandings turtle. Our assay works in both Nova Scotian populations and Ontario populations. And in both cases, we got very nice levels of detection. The wood turtle surveys we just performed last December, and we've got nice occupancy estimates, again from working with local conservation groups who have known occupancy data from conventional methods. In such fashion, we are developing a nice narrative that confirms that eDNA is a less expensive and an efficient monitoring tool that won't surpass conventional methods, but will help triage cases where certain taxa are in dire straits and concentrated conservation and management measures should be implemented more expeditiously. We do aim to continue working on other Ontario turtle species, so if you have one, just give us a ring.
Thank you, Stephen, for answering this question on species at risk turtles. Now we have a question from Zachary Khan. How many amphibian species in Ontario do you already have primers developed for? And related to that, how much does it cost to develop a new primer? I will answer the second part of this question, and then we'll let Steve handle the first part. So developing a new primer can be low cost. And as Mark pointed out, we go well beyond developing any primer. We develop a well-validated assay to ensure the utmost specificity and optimized sensitivity, reaching toward 1 copy of DNA as a limit of detection. So developing, on average, developing a new test fully validated will be around $5,000.
In regards to the first part of the question, I think we had a slide with some of the assays on. If we could navigate back to the slide. Can we do that? Okay, in that case, I'll parlay a list that we have. We have assays for the Jefferson salamander, for the blue spotted salamander, for the unisexual amstomid blue spotted Jefferson complex. We have the Canadian toad, and we have in addition to the Canadian toad, we have the chorus frog as well. Currently, that is, and our final one is the American bullfrog, the Lithobates species. But as Mario says, we can develop an assay for almost any species, given our robust assay development operating procedure.
Thank you, Stephen, for answering the question that was just asked. Now we have Tyler Miller, who is asking what assays are available for aquatic invasive alien species. So again, I'll pass this on to Steve for comments.
All right, thanks for that. We have quite a few assays for potentially and realized invasive species. We have assays for the Dracanian mussel species that are present in Canada, zebra mussel and quagga mussel. We have assays for smallmouth bass, chain pickerel, northern pike, which are invasive in some parts of the country. As I just mentioned, we have an assay for the American bullfrog, which is problematic out west especially.
The carps?
Oh, yeah. So most famously, the Asian carps coming up through the Mississippi watershed and into the Great Lakes. And the New Zealand mud snail as well.
Thank you very much, Steve. Now, a question from Lauren Lawrence from Mi'kmaq Conservation Group in Nova Scotia. So the question is there a possibility that a wood turtle primer based on Ontario populations would not hit on Nova Scotia populations? And if this is a possibility, how would you validate whether this would work or not? Steve.
Okay, that's a very excellent question. When we design assays, we take into account all the within species standing genetic variation that is extant in populations in Canada and beyond where the assay may be applicable. So when we design the assays, we scour the online databases for all genetic sequences from all populations. Where some populations are missing such sequences, we develop our own. We go out, collect tissue samples, and we Sanger sequence them. So we embellish our own databases. In such a fashion, when we're designing assays, we make sure that all the within species genetic variation will be accounted for when the PCR reaction amplifies that target. And of course, this also means that it doesn't amplify any coexisting or co-distributed other turtle species or even other reptiles. So this is for validating our assays in the lab. So obviously we have to make sure. So we do pilot studies in the field. We make sure we go to places where we know wood turtles to be to be localized, and we perform our field surveys in such locations. If we're getting a lack of amplification, we may have to refocus our design efforts and collect more genetic sequence data, because there may be a small risk of a false negative. But in everything we do, we try and maximize the information available to us so that when we do design any assay, it's on the best available knowledge. And if you want to know a little bit more about how we design assays to cover such eventualities, I refer you to our most recent blog post, which is on this very subject.
Thank you, Steve. Now a question from Celia Chui. What is your approach for screening for multiple species at one location? Do you need a priori knowledge of what may or may not be present?
That is a good question. And when we test our assays in the field, in the first instance, we absolutely should have a priori knowledge of what species are present. If we don't have such knowledge, we can't appraise how well our assay performs in the wild, where we know the species to be. In those cases as well, where we have multiple assemblages of potential target species, we also optimize our multiplexing. Multiplexing is performing PCR reactions on multiple species in the same reaction tube. So that information is vital. But essentially, yes, we do require, in the first instance or in pilot studies, known occupancy by the target in some locations.
Thanks, Steve. A question from Brent. Are there any case studies of the application of eDNA technology, specifically in Ontario for SAR fish amphibian turtles? The answer is yes. We have written a number of case studies. If you go to our website, precisionbiomonitoring.com, In the menu, you'll see case studies, and there's a few case studies. The case studies that are there are all from Ontario. We are in the process. We have done a lot of eDNA surveys last year. So we are in the process of writing additional case studies. And we are thinking of having a future webinar specifically developed to present applications and case studies. And again, if you click on the poll button, you can vote for case studies. And the more votes, the more of a chance that we will have a future webinar on case studies. Thank you very much, Brent. Now, Dr. Scott Taroff from Azimuth Environmental Consulting. So Scott is asking, related to the previous question about MNRF, Azimuth ecologists working with Precision Biomonitoring have been in regular contact with about 6 MNRF biologists from multiple region and district offices over the past 2 years. Feedback and support has been generally very positive, and MNRF has requested that we keep them appraised and updated of our sampling process. Thank you very much, Scott, for this important reminder. The next question is from Melanie. Have you investigated the detectability of fish species with or without disturbance to the stream? For example, walking the stream and steering up the fish.
That's a good question. We haven't explicitly looked at that because when we apply our on-site eDNA detection, we purposefully try and avoid getting in the streams and any other water body. The Andy eDNA sampler does come with an extendable boom that extends up to 3.6 meters, I do believe. And at the end of the boom, we have a single-use sampling tube that is completely sterile. It's the only part of the process that will touch the water and penetrate the water surface. So whilst your concern is valid, our approach entirely mitigates against actually disturbing the habitat in such a fashion.
Thank you, Steve. The next question is from David Brown. David is asking, how does PBI's technology differ from the biomeme test kits that Smith Root sells alongside the Andy system? So I will answer this. Our technology differs in 2 areas, and those are the 2 differentiators that Mark has presented earlier on. So the first is how we design our test. We go beyond standards to establish a new gold standard in the quality of our assays, specifically when it comes to true specificity and enhanced sensitivity, and the type of extensive validation we do with third-party and field validation. So this is the first part. We have privileged access to University of Guelph, given that the university was our incubator, and we continue an R&D collaboration with Dr. Bob Hanner at the Biodiversity Institute to keep improving our triple lock method to develop more robust assays. The second main point of differentiation is that, as Mark also explained, the technology for the sampling and the analysis is important. What we have learned is that the study design, the front end, how to design a customized survey for increasing the probability of detection. This is something we have developed here at Precision. We work in close contact with many in the eDNA community, and we are really pioneering efforts in study design. And as Mark also mentioned, we have filed a patent application on the process for survey design, and we are designing a software tool that will enable us to automate the process of study design, and that is part also of the patent application. I hope this answers your question. Next question is from Zachary Khan. Zachary is asking us, has PBI done any work with benthic macro invertebrates? And I will turn to Steve to answer this. The University of Guelph has done work on benthic macro invertebrates. Here at Precision, we are currently considering a project on this. I would point out that benthic macroinvertebrates can be done using a different technique than qPCR, using metabar coding. So we have the capabilities to do this, and we have not been requested to consider such a project. However, if you do have a project on benthic macroinvertebrates, we'd be delighted to speak with you and see how we could work together on this. Steve, just realized that I've answered the question, but I'll let you comment on this.
Yeah, I would say that I do some work still at the University of Guelph, and I discuss these things with a lot of eDNA students there, one of whom is working on macroinvertebrates in certain ecosystems, and he's using a dual approach using targeted qPCR and metabar coding. It seems to him and to me that insects are quite low-shedding organisms. So it may be that we need a multiple approach in terms of these taxa. I think in the first instance, metabar coding is probably more preferable, given the extent that people would like to know about lots of different species within certain taxa, because some are more diagnostic than others about the health of an ecosystem. But yes, we have handled some benthic samples before, and we have metabar coded them. So that is something we do.
Thanks, Steve, for the complimentary comment. The next question is from Celia Chui, and reads, if field samples are needed for validation, for example, from remote areas without existing sequences in bold, can PBI extract and amplify DNA from other tissue types, such as preserved or frozen hair samples, fin clips, etc.? Steve.
Absolutely. We have extracted DNA from empty egg matrix from salamanders and from the mucus from American eel. So hair and fin clips are no problem.
Thank you, Steve. Another question from Tyler Miller. Tyler is asking, it's apparent that study designs have been created for fauna. Are there study designs created for flora? The answer to this is that at present, we have focused specifically on aquatic fauna. And this is because of the demand that we have received from industry and conservation groups. We have not done study design for flora, but I will let Steve complete the answer.
That is a very good question. It is true that we are, quite chauvinist at the moment that we're focusing on animals, but I don't see any problem with extending eDNA to plants. There have been studies in the literature. Canada and other countries are suffering a barrage of invasive plant species, invasive duckweeds, amongst others. Also, when I was in New Zealand, there was an awful alga there that has some disgusting name like snot rot or something. Such things we would be able to develop assays for 100% sure. The sampling design strategy may be slightly different since plants don't have an ability to move themselves unless it's in a gametophyte stage, I want to say. There's also an application for species at risk ferns that do have released free-moving gametes into wetlands, and I think that might be an interesting project that I would quite like to work on if somebody wants to give us a commercial reason. But yes, I believe that plants are amenable to different but equally robust eDNA surveys.
I have a comment here from our colleague Chris Wilson, who is a leading light in eDNA R&D. Chris is pointing out rightly that there are studies looking at aquatic invasive plants in the published literature that include sampling design. So thank you very much for the comment, Chris. This is an area, certainly, when we get requests from conservation groups or environmental consulting groups that we would like to explore further. Thanks. At this moment, there doesn't seem to be any more question. We are at 11:47. If there are more questions, don't hesitate to ask them. Also, if you want to click on the poll button and you can vote on what topic you would like to see covered in future webinars. And I can see the numbers here at 11:48 on January 17. There's a majority of 54%, 62% favoring survey and sampling strategies, and eDNA application and case studies is a solid second. There are some of you would like to learn more about and discuss more about why specificity and sensitivity of qPCR assays are so vital. So I'm going back to the question. Melanie has a question which is the following: do you think there will be an instant detection device developed within 10 years? This is such an excellent question, Melanie, and the vision statement that we have developed for Precision is to be able to detect any species at any time on any spot on the planet. So we are very much engaged with our collaborators to realize this vision of detecting any species anywhere in an instant on the planet. And I hope you can join us in realizing this vision. I, for one, I really think it's possible. And I think that by continuing to work with the universities, with science groups and the government and other companies, that this vision will become true. If you say within 10 years, if you look at 10 years ago was the first ever eDNA publication. So there's been a tremendous progress made in the last 10 years at the point where this is a reality, that it can be done on an industrial scale. And we will work to make this instant detection possible within the next 10 years. So I want to thank you very much for your participation, for your very astute questions, and voting for the next seminar. And I wish you a good rest of the day, and we will be in touch shortly. Thank you so much.